Hi, good evening, everyone. How are you? I hope you guys are doing well. How are you guys? Hello. Can you hear me, guys? Are you able to hear me? Yeah, great. Good evening. Okay, great. So let's start, guys. So we are meeting again for the guidelines classes, guidelines training. And last week I took only two guidelines, and this week I'm planning to take all remaining four guidelines. Okay. So today is the third guideline, and that guideline is very interesting. And I was reading and preparing the slides and reading those guidelines. This is amazing. So I thought like this is something very special, this is something very, very special to teach for me. And it will be very special to learn for you because these guidelines, what we are going to discuss today, are wonderful. Wonderful means the one of the excellent guidelines, very small guidelines, 70 pages, including the bibliography and all, but you will learn a lot. A lot means a lot. So I have added few, the out of the box scenarios here for your learning today, right? So I think you all are aware of the title. Let me share the screen and let's discuss. Yeah. Okay. So today's guidelines, let me make it full screen. Yeah. Okay. Right, guys. Then can you see the full screen here? Can you see the full screen, guys? Hmm. Okay. Yeah. Great. So today's guidelines are nothing but very special ICMR policy statement for the ethical conduct of controlled human infection studies, CHIS, 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 controlled human infection studies. So can someone, the brave participant from this class, tell me what is CHIS? What is Controlled Human Infections Studies? Without looking into the internet, without looking into the Google, can someone tell me what is CHIS? What do you understand by the word Controlled Human Infection Studies? Can someone tell me? Yes? Anyone? Anyone? Anyone can tell me. What is CHIS? What do you understand by Controlled Human Infection Studies? And this is for everyone. See, the people who wants to go to the industry after M Farm, B Farm, or Farm D, or biotech, or any healthcare professional. These guidelines are for everyone. Why these are for everyone? I'll tell you. Okay, specifically works to control the disease. Study to infection disease without letting it break out. Controlled human infection studies. Selected infectious disease patients based on eligibility criteria from control group. Hmm. You all are far away from the answer. Okay. You all are far away from the answers. Okay. So let me tell you the answers. Okay. Yes, 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 yes. It looks like that Sankhanil Panda got the answer. Okay. So controlled human infection studies means we deliberately induce the infection, and we study that infection and the path of the infection, and the vaccines or the drugs to control that infection in a very controlled, highly controlled study design, very highly controlled environment, right? So first we induce the infection in the human beings, and then we study it, and along with that, the drug efficacy or the new drugs or new vaccines or anything, right? So there are many things. Okay, so why it is important for all healthcare people, all technology people also, is because these guidelines talks about the vaccine preparations. These guidelines are talking about the vaccine manufacturing, drug manufacturing for the infections, pandemic, endemic, unknown infections, known infections. So anything, whatever you want to do in the infectious studies, these guidelines are made for that. These guidelines are extension of the last two guidelines. Okay. The majorly the last guidelines, ICMR, ethical conduct of biomedical research, right? So these guidelines are add-on to those guidelines, right? These guidelines came into the existence in 2023, because when the corona came into India, I mean into the world, definitely into the India also. So that was a big chaos in India. Okay, the history behind these guidelines is that was a big chaos in India, and there was no proper regulation. See, studies has been done since long under the different guidelines. There were the study happening, even the chess were happening, controlled human infection studies were happening in India under the different guidelines, under the different regulations, different regulatory bodies, right? It's not like this is something new. Since 1930, 1940, these studies were happening, right, in India and in the world, under the WHO and under the other, CDSC and all, right? CDSC and all. But specifically, just for the, just India has made these statement guidelines, or the guidelines policy statements, in 2023, especially for the controlled human infection studies, right? Otherwise, these were the part of other studies before, our other guidelines is before. Okay, so let's talk about this. Again, I have to check my audience. So there are a few questions to all of you guys. How many of you have read these CHIS guidelines in India? Like, how many of you have studied the CHIS to 2023 guidelines? Yes, guys, answer fast. I'm expecting more answers because people are from industry also. No one? Anyone who studied or who read these guidelines before? No? Okay. Okay, no one, nobody's. No, that's perfectly fine. So all other answers are just, the things what we are going to discuss then. See, so the next question is, give me two major glimpse. No idea. What is deliberate infection? Okay, can someone just guess what is deliberate infection? I mean, there are the four terms I kept here. Can someone tell me one by one? Let's talk about one by one. What is deliberate infection? Can someone tell me what is deliberate infection, guys? Yes. What is deliberate infection? Okay, let me open your mics also, isn't it? So you will speak. Yeah, you will be able to speak. Yeah, guys, you can speak. Yes, tell me, what is deliberate infection? You can speak or you can write, anything is fine. Infection made consciously, getting infection on purpose of the study, inducing infections in a person, infecting a participant on purpose. Yes, deliberate infection means we have a purpose of the studying either the infection path or the drug. Okay, so for that we usually induce the infection in the human being, right? With a purpose, we are inducing the infection in the human being. That is called as deliberate infection. Deliberate infection. Can someone tell me this fancy word, ethical dumping? And this you will learn a lot and you will hear a lot when you're going to industry. Okay, when you're going to industry, you will hear this word a lot. In institutions, we don't hear this word ethical dumping. But when you're going to the industry, you will see a lot of people talking about ethical dumping. Okay, so can someone tell me what is ethical dumping? Just without looking into the Google, I'll tell you everything. Okay, I'll tell you everything. So just whatever your conscious say, you just write that. Okay, whatever your conscious say about these terms. Yes, guys, ethical dumping. Can someone tell me about ethical dumping? This is for everyone. These guidelines are for everyone. No one is out of these guidelines. Yes, ethical dumping. Can someone tell me ethical dumping? Maybe something which has a lot of, Ruchita is telling, maybe being unethical. Okay, Neha is telling being ethical. Ruchita is telling maybe something which has lots of ethics. Okay, both are the exactly opposite statements, followed by regulatory bodies, etc., for a proper study. Okay, okay. Mm-hmm. Yes. Anyone else wants to put there? Followed by regulatory bodies, etc., for a proper study, and then okay. So I think you guys are not up to the the point here. Let me tell you. Let me tell you. This is a very new word. This is a very new word and very specific word, because you are being the student or being the very beginner into the industry. You might not be knowing this word. Okay, you might not be knowing these words, right? So let me tell you what is ethical dumping. See, ethical dumping states whenever we do. See, there are two types of countries, right? One is developed countries. Developed countries, right? Like USA or some European countries, right? European countries like maybe UK, you can take Spain, right? Or you can take Germany, you can take Japan, right? And there are underdeveloping countries, under developing or developing countries, right? Like India, African countries, right? African countries, South Asian, some of the South Asian countries like, Nepal, Sri Lanka, Pakistan, Afghanistan. Or these are the some of the European countries also, some of the, US states also are there, but the main part are these underdeveloping states or underdeveloping countries, right? So what is ethical dumping is, there are two types of guidelines also. There are two types of guidelines for the research. There are two types of guidelines for the research in these two types of countries. Okay. So here, the guidelines which are made very strict. Okay. For such studies like chess. Okay. Such studies like chess or any drug development studies, the guidelines, the ethical guidelines are very, very strict and hard. But these guidelines are completely broken. Okay. These guidelines are completely broken and completely avoided in underdeveloping countries. Okay. Are completely, completely avoided or broken in these underdeveloping countries, just to test the infections or just to test the drug which is harmful and which cannot be tested in developed countries. Understand? So that is called as ethical dumping in such states or in such countries where the guidelines are avoided completely, or the research is doing forcefully, or for the need of the people. These people need the money and they are, being the guinea pig for the clinical research. In such countries, these guidelines are completely avoided because their financial status is not good, because they are lacking of the finances. So the countries such as US and the developed countries do not study. You listen to it carefully, guys. They do not study such drugs or such infections or such controlled human infection studies in the developed countries, but they do it in the underdeveloping countries or underdeveloped countries, because their guidelines are completely broken. Their guidelines are completely avoided. Okay. So this term is called as ethical dumping, because you can do those things easily which cannot be done in the developed countries easily. Understand? Understand, guys, what is ethical dumping? So it is happening. It is happening. Even the international guidelines, international uh, the rules and regulations for clinical research are avoided in such countries, completely avoided, because they need finances, because they need money. So the people do anything for the money. Even they participate in such studies which are completely banned in the countries such as US and Japan and Germany. But those studies are being done in Kenya, or the Guinea, then Papua Guinea, or such as some other African countries. And those studies are being done in such other countries also, like India, Pakistan, and all, right? So these studies can be done in such countries. That is called as ethical dumping. Understand, guys? Understood or not? Did you understand ethical dumping? Yes, tell me. Did you understand it, or you want something else to understand in this? Great. Okay, so that is ethical dumping. What do you understand by stigmatization? Tell me, what is stigmatization? My electricity is off, so you cannot see my face clearly right now. That's okay. Yeah. What is stigmatization? Yes, tell me, guys. So, sir, is it allowed in other countries by people from other countries? Yes, for money, definitely, because US is a country who gives the funds to the other countries for survival. Okay, and UN is United Nations is in America, and FDA is in America, CDC is in America. So everything is fixed. For such studies, everything is fixed, and the countries, the underdeveloped or underdeveloping countries, they need the funds. Okay, they need the funds. That's why they allow, and they made the guidelines in such a way that everyone can come and do such studies, or their countries can these countries can do the studies like this for other international, because see, if you look at the clinical trials are not the the single nations. All the clinical trials, vaccine trials are happening in all the nations all together, right? So those are called as multinational, multicentric studies. So just at the by the name of multicentric, national, they just do those clinical trials mostly in such countries where those are easy to do. Understand? So now tell me, what is stigmatization? What do you understand by stigmatization? You're going to learn a lot of new things in these guidelines, guys. Okay, stigmatization is kind of clouding of judgment due to fear or false information. Okay, treating in a negative way. Okay, okay, yeah. Or something else? Anyone wants to put some more lighting, enlighten the word stigmatization? Yes, guys. What is stigmatization? Okay, let me tell you what is stigmatization. So, suppose I'll give you an example. There is one person. There is one person who has the different characteristic, either one or maybe all. Maybe the different race, maybe the different cultural values, right? Maybe the different cultural values, maybe the different color of skin, okay? Maybe the different religion, or maybe the different whatever the people can differentiate a person for. Okay, maybe the political thoughts or whatever you think, that the list is a long list. Okay, so race, cultural values, or the skin color, or anything, it could be, right? It could be anything. So, and the population, second thing is population. His thoughts, his race, his cultural value, his color, skin, or anything is not matching with the population. Okay, so people make a judgment about. Okay, people make a judgment about this person, him or her. Okay, and they just make it in the completely negative way. They stigmatize this person. They just completely discriminate. Okay, discriminate and differentiate the feelings, the logic, the thoughts of the entire population against this person. Okay, so in the clinical trial, that is called stigmatization. In the normal life also, it is called stigmatization. Okay, but in the clinical trial, the stigmatization word is used to some specific person or specific participant. Okay, like, okay, we are having some 4 stigmatized patients or participant in this clinical trial, who are representing such population, who are representing such race, who are representing such cultural values, who are representing such religion. Okay, so that is called as stigmatization in the clinical research. And it is happening in the ethical, it is happening in the chess, because chess are the most important type of studies, right? Infection control, infection development, infection, because see, you have seen the corona, how corona spread over the world, and it was devastating, right? It was devastating. It has almost more than 5-6 million deaths all over the world, right? 8 million deaths, I think. Not 8 million deaths, 5 million. How much deaths? I literally forgot. 50 lakhs deaths, right? Something all over the world, right? So 5 million deaths all over the world. So such studies can be done. Such studies must be done to tackle such programs. The such pandemic or endemic situations, right? So that's why they, on the name of such clinical trials, on the name of such clinical trials, they, stigmatize the peoples and their feelings and their, all the cultural values or race, right? So these are the specific things. These are the specific things, and that is called as stigmatization in the normal life, also in the clinical trials also, right? What is test of understanding? Can someone tell me test of understanding? You are very much familiar with this word. Okay. You are very much familiar with the test of understanding. What is test of understanding, guys, according to you? Tell me. Yes. Test of understanding. Yes, guys. Fast, fast. Do you know about the test of understanding? What is test of understanding? Yes, everyone? Don't know? Okay. Anyone who wants to just say about test of understanding? Patient history. Okay. To know the ability. Okay. To check whether the info is known to someone. Okay. Done maybe by asking questions. Okay. Ruchita, somehow you are relating to the answer. Okay. So test of understanding is very easy. You all know about the test of understanding. See, one participant is participating in any clinical trial. So we take what? We take the informed consent, right? Informed consent. Informed consent means we are informing him all the information, all the information about clinical trial, compensation, risks, benefits, whatever, all the information we are giving. So just before he signs or she signs the IC informed consent, we take a test. Okay, we take a verbal test with some 5 to 10 questions. 5 to 10 questions. We either take a verbal test or through the Google form or written test, maybe multiple choice questions or one word answer question. That test is called as test of understanding. Okay, how simple it is, and you guys don't know, right? But it is very simple. Definitely the term is new for all of you, I understand, right? So this is like, if the person has understood, if the person has understood the information you have given to a participant, if the participant has understood the information whatever you has given. Okay, so that is called as test of understanding. Very easy to understand. Okay, understand? So shall I take a test of understanding? Okay, let it be. So guys, let's talk about the CHIS guidelines, controlled human infection studies guidelines. Okay, so first of all, the CHIS guidelines involve a research design that intentionally exposes healthy human participants to a specific pathogen or infectious agent. Okay, you understand the word. This is the word, intentionally exposes the healthy human participants. Intentionally exposes the healthy human participants. Okay, to a specific pathogen or infection agent, either in a wild type, its wild type, or in its attenuated form. Okay, can someone tell me what is the difference between wild and attenuated form of an organism? Yes, fast, fast, guys. What is the difference between wild and attenuated form of an organism? Yes. So they do such studies, right? Under the controlled conditions, following stringent, like very tough protocols, right? Active form and inactive form. Absolutely correct. Active form and inactive form. So wild type is active and attenuated form is inactive form. Inactive form, right? So they synthesize the virus DNA and they use that DNA, or sometimes nowadays mRNA also is being used. Okay, so active and inactive form of the virus or bacteria or any organism. Okay, or any organism, that is called as wild type or attenuated form. Understand? So the summary of this is research designs that intentionally exposes a healthy human participant to a specific pathogen or infectious agent, either active or inactive form, under the controlled conditions following the stringent protocols. Okay, why we exactly do that? why we exactly do that? To assess the vaccines, or to assess the treatment modalities and accelerate the research through enhanced insights into the disease process in the human, right? To check the vaccines, I mean, to assess the vaccines or treatment modalities, and to check the disease process in the humans. Okay, right? Understand, guys? So these are very important and very specific, because there are the things like you have to assess the vaccines or treatment modalities or access the, you know, disease process or disease path, how it is happening, what are the system involved, what is the process of, genesis of the virus, and everything you will study in the in such, such kind of studies, right? In contrast to lengthier natural infection studies, in contrast to lengthier natural studies, these studies may offer the advantage. Guys, listen carefully. These studies may offer the advantages of quickly obtaining the data and delivering accelerated results with smaller sample size. So these, why these studies are important, right? First of all, we are assessing the vaccines, treatment modalities, we are checking the disease path. And other than that, these are quick studies. These are quick studies. So see, sometimes pandemic is very longer. Okay, pandemic is like, 6 months, 1 year, 2 years, 3 years. Corona has come, corona has come down in 3 years after three waves. After three large waves, the corona has come down in 3 years, right? So such a lengthier infections are there in the nature, right? So to tackle those and to, get it ready to tackle such infections, we have the quick settings. Like quick settings means the quick environments where we can test such infections or, their treatments, vaccines and all, right? Understand, with a lower sample size. So if we have benefits, we must have some kind of problems also. But before we go to the problems and disadvantages, let's learn more about these. So CHIS conduct requires some, there are some requirements for the, controlled human infection studies. Those are, it should be more careful, and it should be under the deliberate review process of the protocol about the ethics, and there should be like ensure local relevance. Like, pandemic and endemics are not the nation, sometimes not the nationwide or globally, they are, they are the at the small levels also, right? Not the pandemic or something, but those are at the small levels, like in, infection is spreaded in specific states or specific places or specific, cities sometimes. So to, it must be ensuring the local relevance, right? The review process must be very, very comprehensive, and these should be under the controlled environment more carefully. It should have the public benefit definitely, because if you are inducing the infection into a human being, that should be with the public benefit, or that should be completely with the local relevance and fair processes. Okay, rigorous ethical oversight must be done with all possible safeguards to protect the human participant from harm, right? So CHIS conducts require such things which you should keep in mind: more careful, deliberate review process, ensuring the local relevance, public benefit, fair process, rigorous ethical oversight, and safeguard to protect the study participants. Right. So social value, what's the social value out of it? What is the social value out of these guidelines? First of all, India is suffering. India has the mortality and morbidity rate very, very high because of the infectious diseases, very high mortality rate. If you understand, the 30% of the total disease burden in the country belongs to the infectious diseases. Okay, belongs to the infectious diseases, right? So what is, there are two specific, departments in ICMR. One is NCD, one is CD. Can someone tell me what is NCD and CD? Yes, guys. Yes, there are two specific departments in ICMR to tackle all the guidelines and problems. What are those? NCD and CD. What is CD and NCD? Non-communicable diseases and communicable diseases department. So the guidelines, or I mean the epidemiology says that 30% of the total disease burden in India is because of the communicable diseases. Okay, communicable or infectious diseases. Okay, communicable is a wider disease, and communicable diseases are mostly all the communicable diseases are either virus borne or bacterial borne. Okay, either virus borne or bacterial borne. Okay, so in Indian context, what is the social value of this? You have to understand. CHIS could be a valuable research tool in bringing effective interventions. Effective interventions for what? Reducing the disease burden in a timely manner, like we did for Corona. India was one of the top country to control the corona within time with its vaccine development, with its proper monitoring and vaccine development, proper monitoring and proper data collection. Everything was very proper in India, and we tackled corona very well. Okay, and we helped almost 124 other countries in such a in such a bad time, right? Our application for corona, the COVID, there was an application, and there was the vaccine development, the vaccines. We helped 124 countries, and we were one of the most important country and most successful country in controlling the corona. So because of what? Because of the faster process faster process of clinical trials, because of the fair process of the clinical trial, and because of the timely distribution of the vaccine and tasks. Okay, these were the three things what was done by ICMR and other government departments in corona vaccine making. Okay, and that was the one thing which helped in control these kind of disease burden in India in such a big population. Why India is important country? Because in such a big population, if we are controlling such pandemics nicely, definitely there was the deaths, and that toll was really bad in India itself, because India has the larger population, right? Almost largest population in the world. So these things can be controlled with the you know with such guidelines and with such processes. Okay, so first, reducing the disease burden. Then CHIS can add the social value in supporting the public health responses to the disease of concern. Okay. Then improve the pandemic preparedness. Yes. So we did we learned a lot of lessons from the corona, and now we have a lot of preparedness for such other upcoming, if there are any pandemics, like if some pandemic happens in Kerala. We have heard a lot of new cases of other virus infections and bacterial infections in Kerala, or in Saudi, or in South African countries. So a lot of infections are going on all over the world. So we are prepared because of such studies in our laboratories, in our the populations, because of such guidelines we have. Then community empowerment. If our communities are infection free or communities are disease free, they are empowered, right? Then we have the economic advantage, because if we are always into the disease progression, disease burden, then it spends a lot of funds also, right? If we are prepared well, if we are tackling well such infections, then we are economically strong, right? And again, we are assisting the timely healthcare decision making. So CHIS guidelines have a lot of social values. That's why I was telling, all the other guidelines is really good. They are thinking about the participant, they are thinking about the safety, well-being of the participants. But why these guidelines are important is because these have a lot of social values, because these are directly affecting, these are directly affecting our community, these are directly affecting the pandemics, endemics, and they're reducing the disease burden and all. Understand? So clear, guys? Let's go to the next slide. Now, limitations of the CHIS guidelines. Can someone tell me, till now we have discussed a lot. So what we have discussed, based on that, can someone tell me what could be the limitations of CHIS guidelines? What could be the limitations of the CHIS guidelines? Can someone tell me? Yes, everyone. Yes. See, from the advantage only you can make out the disadvantages. From the advantages only you can make out the disadvantages. Please tell me. It's not universal. Okay. causing further complications in the patients, selection of the participants, lead to adverse effects later. Okay. Okay. Few are correct, few are not correct. Let me tell you, ADRs. Okay, so let's check. Let's check what are the limitations. See, first of all, you must go back and see what are the advantages. What was the advantage? Number one, quick studies. Number two, small sample size. Can someone tell me what are the disadvantages now? Can someone tell me what are the disadvantages now? These are the advantages we studies. Quick process, small sample size. What are the disadvantages now? Let's check your cognitive power, guys. Yes. See, you are the graduates. Okay. You should know that. Maybe biased. Okay. See, answer is written on the screen. Not necessary. Ah, fantastic. See, not necessary to work on all due to smaller sample size. Absolutely correct. A small sample size did not give the overall results of the larger population. Not applicable for other populations. Absolutely. Since the sample size is small, result might not be applicable. Yes. No much time for not monitor or whatever, but why the small sample size is because it cannot represent the entire population. First of all, quick studies means in the quick studies, we cannot check the longer prognosis. We cannot check the long prognosis of the disease. We cannot check the long effect of the disease in the people. Like, have you heard of one term? I mean, all of you must have heard of. All of you must have heard of, right? What is this? What is this? Can someone tell me? Long COVID. What is long COVID? Just define the long COVID in one line. Anyone? What is long COVID? Yes. What is long COVID? Yes, the sign and symptoms of the COVID persist for the longer period of time. Not exactly, but no, long COVID means not at all recurrence, not at all duration of the COVID, not at all persistent, longer time of period. No, 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 no. Long COVID means the COVID related complications, the COVID related thrombotic disease, COVID related new onset of diabetes, COVID related new heart attacks or MIs, COVID related new autoimmune diseases. Okay, so COVID has affected each and every cell of the body, each and every system of the body. So those things are showing the impact or showing the problems the later stage after 1 month, 6 months, 8 months, 10 months, 11 months, 2 years also. Okay, so that is called as long COVID. That is the term is given as long COVID for it. Okay, there are multiple complications related to the COVID. Okay. And these have increased drastically. The new onset of diabetes has drastically increased in the kids and the elderly people after the COVID and myocardial infarctions. You must have seen a lot of articles on COVID-induced thrombosis, COVID-induced thrombosis, COVID-induced hemorrhages, COVID-induced multiple things are there. So that entire complications which are associated with COVID are termed as long COVID. Okay, these things are termed as long COVID. You must study about the long COVID. Okay, so let's check about the limitations of the long COVID. See, just come up with a set of challenges and limitations, mainly with the safety of participants being a major concern due to the nature of risk involved to the study participant. Definitely, if you are giving such a new organ or organism or virus or bacteria, whatever, that could be risky. You don't know what could be the risk, what level of the risk you are taking with the participant, first of all. Then data produced from CHIS may have limited generalizability, due to the testing carried out in highly controlled studies, highly controlled conditions, See, whenever the corona came, was there any controlled environment? Not at all. corona was free of everything. Okay, it was free of everything. It was spreading in the air. It was spreading on the walls. It was spreading on the iron rods. It was spreading on the books. It was spreading everywhere. Okay, it was spreading everywhere and through every almost all the modes, right? So there are no controlled environment when pandemics and endemics are happening. Okay, there are no controlled environment when pandemics and endemics are happening, right? So we don't, we cannot just say that highly controlled conditions, testing in the highly controlled conditions can replicate in the original conditions or the natural conditions. First of all, this. Then the small numbers may not represent the general population. Understand? Thereby, it lead to a bias in outcomes, right? Then it may provide the short-term data, making it challenging to assess the long-term effects of infections and treatments in the people. Like first of all, the long COVID, and then vaccine-related problems, vaccine-related thrombolytic events, vaccine-related thrombolytic events in the people, right? So the COVID vaccine also caused some problems. Either those are 0.001%, but there were the problems in the people, right? So such things could be the limitations or the challenges in case studies. Then use of attenuated pathogens or specific well-characterized strain selected for safety and tested in the laboratory testing. Testing may not accurately represent the behavior of wild organism, right? So you cannot expect that the inactive form will behave like active exactly. You understand? So there are these major and logical limitations. Understand? Logical. First of all, nature of the risk to the participants, then the limited generalizability, then the small sample size number, right? And then short-term data, right? And then the attenuated pathogens. These are the five major limitations of the CHIS studies. Okay, major limitations of the CHIS studies. And so guidelines also cannot help in that, right? Right, understand guys? Clear? Clear till here? Anything else? Guys, clear till here? Okay, great, great. Thank you, thank you. Okay, so now the next thing comes here is why we created guidelines for ethical conduct of chess. Why we created guidelines for ethical conduct of chess? Can someone answer this question for me? Can someone answer this question for me? Why we created guidelines for ethical conduct of controlled human infection studies? Yes, guys. Patient safety, perfectly fine. Patient safety, because human participants are everywhere. Human participants are everywhere. So they have the specific objectives. What are those objectives? Let's learn. What are the objectives? These are the objectives. So first is to identify the ethical considerations around the conduct of chess while ensuring the protection of safety, rights, and well-being of research participants. First thing is why we have ethical guidelines for chess is to protect the safety, rights, and well-being of the research participants. Okay, that's why these guidelines are made. Then to provide a framework to the ethics committee involved reviewing the chess. So there are a little different guidelines for the ethics committee and ethics committee members also in the reviewing of chess, right? Just to ensure the safety, right, and well-being of the participants. Understand? So these are the specific criteria. These are the specific objectives. These are the specific objectives for ethical conduct of chess, right? Now let's come to the main point. Stages of chess studies. Stages of chess studies. Can someone, tell me what could be the stages of chess studies? According to you, just, what is chess? Controlled human infection studies. What could be the stage? I have brought wonderful examples for you out of these guidelines today, right? Because these, literally touched me. These guidelines literally touched me and I read multiple cases. Okay, multiple case scenarios I read about the chess studies and then I made these things for all of you guys, right? So tell me, what are the stages of chess studies? Anyone, like you can throw anything. I'll catch everything. Okay. Any Wrong answer will be the answer here. Consent testing analysis. Okay. Anything else? Anything else, guys? Consent testing analysis. Perfectly fine. I mean, these are the major steps. These are the major steps of research, Ruchita, not the stages of CHIS studies. Okay. These are the but major steps of the research. Selection of healthy participants. Okay. Anything else, guys? Yes, people. Yes. Obtaining IPS. Okay. Let's uh, check. Let's check what is this. Okay. Let's check what is this. Then we'll go further. So, CHIS are conducted for testing, therapeutic, or vaccines, typically in two stages, also considered as two type of studies. Two stages of the CHIS studies are also known as or also considered as two types of studies. Okay, so you are definitely not knowing those stages. I'll tell you. Okay, so first stage. First stage is very simple. Whenever you are doing such studies, you will first induce the infection. You will develop the infection. Understand? You will first develop the infection. You understand? First stage is development of the infection, and the second stage is treatment of the infection. Simple or not? Isn't it simple? Stages. First stage is controlled human infection development. Controlled human infection development studies. That is called as what? That is called as CHIM. Okay, CHIM. CHIM is controlled human infection model. First, we have to develop the controlled human infection model. First, we have to develop the controlled human infection model. So first stage can be called as CHIM. First stage can be called as CHIM. So in the first stage, when testing a new drug, healthy human volunteers are given the wild strain or attenuated pathogen in order to induce an infection or disease, usually mild or self-limiting or easily treatable. Okay, whether you are giving a mild or the attenuated form of the organism, it should be easily treatable, self-limiting, and it should be mildly affecting. Okay, we cannot take the wild form, means the exact natural wild form of any organism when we are testing it. So we induce the infection, and this process is known as development of the controlled human infection model, CHIM. Okay, development of CHIM for that disease. Okay, for example, there could be the CHIM for tuberculosis CHIM, typhoid CHIM, right? Typhoid CHIM, HCV CHIM. Human. What is this HCV? What is HCV? Hepatitis C virus. Okay, hepatitis C virus CHIM, malaria CHIM. So anything that's called as CHIM. Okay, so CHIM studies are very, very important. Development model, right? Development model. Yes. So here are some examples. Okay, here are some examples for all of you guys. I have brought these examples. I studied these complete papers. These are the complete papers and published in JAMA, Nature, and all. I studied many papers for all of you, literally, and these were so interesting. Okay, how they did these CHIM studies and how they prepared these models and all. This is wonderful. So they made the models, right? So this is what? This is inhaled BCG controlled human infection model. This is for BCG. What is BCG? Can someone tell me? BCG. What is the full form of BCG? Yeah. Anyone? Yes. BCG. Come on, guys. This is very simple. BCG is the combination. Okay. Bacillus, calmate, varin. Okay. What does it exactly mean? What is it exactly? So this is the attenuated form. This is the attenuated form vaccine. Okay, for TB, right? This is the attenuated form vaccine for TB. See, So first they give the vaccine. Okay, this is not exactly the vaccine, but this is exactly, I mean, not the vaccine, but the combination of the tuberculosis bacteria. Tuberculosis bacteria or virus, guys? Can someone tell me it is a bacteria or virus? Sorry. Yes. Is it a bacteria? Yes, tuberculosis. Okay, so they gave the BCG nebulization to the patient, healthy subject, and they did the bronchoscopy to check the alveolar air space. Okay, to check the alveolar air space, and from that they received the fluid, and then they did the quantification. Quantification. How they did the quantification is like, first see, they recovered it via bronchoscopic saline levels at the defined time points, and then they checked how much it is affecting in the next stage. So this was the developmental first stage. Okay, it was the developmental first stage of the BCG model, BCG chim. Okay, how they developed the BCG is they took the BCG attenuated form, they nebulized to the healthy subject, and then they did the bronchoscopy, and like bronchoscopy with what, with the saline levels. They took the saline and from the air spaces, alveoli, and then they quantified that. Then they quantified the BCG. Okay, so this is the BCG controlled human infection model, human development model. Okay, now next I'll show you the typhoid controlled human infection model, typhoid chimp. Okay, so in the first stage, they recruited the volunteers at the baseline. They recruited the volunteers. Then they challenged with Salmonella typhi quails strain. Okay, they did it with the Salmonella typhi, and what, two doses. High dose at 1 to 5 into 10 to the power 4 cells, and then low dose 1 to 5 into 10 to the power 3 doses. Almost, these studies were done at a 10x. Okay, at a 10x, 1 to 5 into 10 to 3, 1 to 5 into 10 to the 4. Studies they did on the 10x, 0x, 0 and 10x. Okay, so very high CFU they did. Okay, it was day one, or sorry, day zero. At the day zero, they challenged with the S typhi. So here in the next stage, at the day 7, they checked for the outcomes. So few people developed TD. TD is volunteers who developed the typhoid disease, and no TD is volunteers who did not develop the typhoid disease. So TD and no TD. Then they observed at these were the different, what we say, the cell types of that. So after day 7, they post challenge, they observe the patient every week, day 14, day 28, until 1 year, day 360. They check the patient till day 360. Okay, so here is the full process. Volunteers were recruited and challenged on day 0 with Salmonella typhi at earlier of 2 doses. At around 7 days, some of the volunteers developed typhoid disease, while others did not develop the typhoid disease. On day 14, all volunteers received antibiotic. On day 14, all volunteers received antibiotics. Okay, and then they followed for 360 days after challenge. Okay, so definitely at D14, D18, there was no TD. Everyone was no TD. At D60 also, there was no TD. Right, so that's how the typhoid was created. This is not only the one stage. Here in the typhoid controlled chimp, here in the typhoid chimp, they showed both the stages. First, like the, here they showed the chimp also, and here they showed the next stage also. So how you can differentiate in the two stages? Here, see, till here, this was the first stage. What we are talking about. Till here, this is the stage 1, chimp. And here stage 2, the challenge. Okay, here stage 2 is challenge. Okay, stage 1 is development of the organism. Stage 2 is the challenge. Stage 2 is the challenge. Understand? So in the challenge, they administered the antibiotic, and then they followed up at different weeks, and they found there was no TD in the participant. There was no TD in the participants at all, right? So that's how they developed all the models. This is just the basics. You must read all the papers, okay? So, and there are some risk mitigations for the chim studies. Definitely, there are some risk mitigations when, the chim studies are being done. As I told you, initially, I told you about something that is called as ethical dumping, right? So to stop or prevent the ethical dumping, there are some risk mitigation policies which should be done. The research institutions must be having the specialized training for the research staff, isolation unit, and 24/7 access to the healthcare, and appropriate biosafety precautions for the CHIM studies and collaborations among research institutions. General public must be advocated for such risks. Okay, general public must be advocated for such risks, like they are participating in such a dangerous, if I say dangerous studies, but that is good for the society. They are doing something good for the society and getting some benefit, so they must know their rights. Okay, the public must know their rights and public must know about the study. Then the community engagement is very important. Then involvement of the social media, collaboration with the government partners for the general public advocacy that can be done here. Okay, in the regulatory bodies, they should have the specific regulatory framework. They should have the revised compensation rules according to the country and according to things, and comprehensive institutional insurance things, so they should be having, right? And then accreditation of institution and ICs with the national committees, with the national regulatory committees. It must be there. Okay, it must be there with the national regulatory committees. Okay. Okay, yeah. So, and then the ethics committee, institutional ethics committee must be trained, specialized training for the ethics committee members, empowerment of the ethics committee, and then close monitoring over the consent and recruitment of the patient. There should be no selection bias. There should be no selection bias. No selection bias in CHIM studies. Okay, so that's about the CHIM studies. Now the second stage is here. Okay, the second stage is here. Stage of the CHIS studies. The first stage was CHIM. The second stage is controlled human infection testing studies. Okay, usually call it CHIT. Okay, so CHIS has two stages. One is CHIM, one is CHIT. Okay, one is CHIM, one is CHIT, right? So these are the two stages. Second stage is the controlled human infection testing studies. So here at the second stage, the participants are randomized, often with double blinding. Even the investigator is not knowing, even not the participants. No investigator or participant is knowing about the drug, whatever the drug is being given to the participants. Okay, no one knows. Participants are randomized and with double blinding, often double blinding in the testing stages to receive either the new drug or currently available drug if no treatment is available as a placebo, right? So either placebo or the other available drug can be used in such things. Okay, so these two arms are then compared to the test the pathogen clearance time. Okay, pathogen clearance time. Can someone just explain what is pathogen clearance time? What could be the definition of pathogen clearance time? Yes, guys. So in the testing studies, we check for the pathogen clearance time using the molecular biology techniques or drugs capacity to treat the infection caused by the challenged strain. Caused by the challenge strain, we check the pathogen clearance time, like in how much time the pathogen is cleared from the body. Okay, from the participant. Okay, from the participant's body, how much time it is there. Time required to eradicate the pathogens, definitely. Okay, time required to clear out the pathogen from the body. Okay, eradication of the pathogen. So this is called as CHIT studies. What there are the examples? See, this is the trial testing designed for malaria. Okay, CHMI. Okay, CHMI, controlled human malaria infection. Okay, this is the controlled human malaria infection study. Okay, you can check in the Google. It's a wonderful paper, lovely paper. Literally, when you will read it, you will, come to know about all the stages, everything, whatever they are done, and amazing results of this study. CHMI study. Okay, CHMI study. So trial testing designed for malaria. So controlled human malaria infection. Okay, so when we are testing the medicine or vaccine, so there are two stages. I mean, there are two types. Okay, guys, there are two types for CHMI. The CHMI stages, CHMI was done in two stages. So what is first stage? I mean, two types you can say. In first, if we are testing the vaccine or if we are testing the medicine. Okay, if we are testing the vaccine, if we are testing the medicine, there are two different approaches. Okay, to test the vaccine or to test the treatment medicine, there are two different approaches. If we are testing the vaccine, then first we randomize the person. I mean, when we randomize the people, then we immunize them to the malaria infection, and then we induce the infection, and then we check the protection time. Okay, then we check the protection time. First, we are randomizing. In the vaccine cases, usually all the vaccine testing happens like that. First, we are randomizing the people, we are immunizing them, and then we are inducing the infection, and then we are checking the protection time. How much time the person is protected from the infection with effect of vaccine. Understand, with effect of vaccine, how much time the participant is protected. And that is being done in very controlled area, very much controlled area. What controlled area? Controlled area means, like, if we are risking the patient with such kind of immunization or infections, we must admit them in controlled approaches or controlled settings, like the hospital settings with the isolation ward and everything. Then we do such studies. If the things are not that not in control of the investigator or the researcher or the sponsor. So these things must be done in the controlled environment. First, for the vaccine, first randomization, immunization, then infection, then we check for the protection. Okay, so here, when testing the vaccine, two groups of volunteers will be randomized, immunized with the placebo and vaccine, and or the comparator subsequently infected with malaria. Endpoint in such trial is blood stages pericythemia, which may not occur full protection or to be reduced partial protection. So in this, there were two things. Okay, definitely randomization means, see, one is control, one is with immunization. Okay, and then after infection, we checked for two things. Either it was fully protected or partially protected, right? Or either it was pericythemia was completely protected or partially protected. So outcome was looked in two stages. One, either it was fully protected by the vaccine or partially protected by the vaccine. This is the protection with vaccine. But when we are checking the medicine, when we are checking the drug, first we induce the infection, then we randomize, then we give the treatment, and then we check for the clearance. Okay, here we are not checking the protection time, here we are checking the clearance. So while testing the novel anti-malarial medicines, volunteers will be infected and subsequently randomized to receive the novel medication or comparator drug. Okay, and then the end point outcome will be the parasite clearance time. And the comparator and the vaccine, or I mean comparator and the drugs will be checked, will be tested in such cases. You understand this, guys? If you understand this or not, shall I repeat it? Tell me. It's heavy raining, so my voice will be quite disturbed. Yeah, guys, tell me. Do you understand it or not? Do you understand it or not? Yes, guys. You understood or not? Just tell me. Yes, everyone. Yes, guys, tell me. Please repeat. Okay, see, I'll make it very simple. I'll try to make it simple now. There are two things. One is vaccine, one is medicine. Okay, one is vaccine, one is medicine. So in the vaccine, the randomization part comes first. Like first, the patients are randomized. One will take the immunization, like one will take the vaccine, one will take either placebo or the comparator drug, vaccine or comparator drug. Then both the groups will be, like exposed to the infection. Both the groups will be exposed to the infection. And then we will check the protection time. Protection time means, like, person is already vaccinated or the comparator drug or placebo is given. So we will compare these two groups, like in which group the protection is much, like either it is fully protected, like fully protected by the vaccine, or partially protected, what is the protection time? Okay, what is the protection time? Like completely, there is no effect of the infection in the body. Completely, that is called full protection. When there is no impact of infection at all in the body, that is called as complete or full protection. Then the second thing is partial protection. Like, okay, the person might not be getting chills with malaria, person might not be getting admitted with malaria, but he is getting a little fever. Okay, he is getting a little chills, or he is getting the partial symptoms, not the complete sign and symptoms of the malaria. Okay, so these type of protection time and protection impact will be checked. So this is a very simple criteria. First, randomization of the people into the controlled and the vaccine. Then we give those vaccines, and then we expose to the infection, and outcome differences. Like here we check the protection. What is the protection? Fully protected or partially protected? And we compare both the groups to get the efficacy of the vaccine. Okay. But in case of medicine, what we do is first we induce the infection. First we induce the malaria. Here we are talking about the testing design of malaria. So here we are inducing the malaria first. Then we are dividing, the randomizing the patients. Randomizing the patients. Now people are infected. All the people are infected here. Okay. All the people are infected. Now we are randomizing them to either control or the test drug. Okay, either control or the test drug, control or comparator. And then we are checking the time of clearance. Time of clearance means after giving the drug, how much time the drug or comparator is taking to clear the infection from the body. You understand? Clearance time means how much time the drug or comparator is taking to clear the infection from the body. That is called clearance time. So they're very simple. The process is different and outcome is different in both the trial designs. In both the trial designs, one thing is process is different. Like randomization in vaccination will be done first. In medicine, it will be done after the exposure. Okay, then outcome first. It is protection. Why protection? Because infection is given later. Okay, infection is exposed later, so we'll see how much time it will be protected. But in case of the drug treatment, the drug trial, we are checking the clearance time because there is the infection before itself. Okay, we are exposing the patient or participants initially, and then we are looking into the clearance time, right? So these are the two types of trial testing designed for malaria. Understand, guys? Now clear? I hope it is clear now. Okay, great, 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 great. Thank you. Okay, so let's go further. Again, very important, very important bacteria, very important bacteria. That is enterotoxigenic Escherichia coli, ETEC CHIM studies. Okay, ETEC CHIM studies. So again, these are the vaccinations for, Escherichia coli, Escherichia coli. So first, as this is vaccine, first randomization is done. First randomization is done. Then vaccine is given. Then the challenge was given, right? Then the challenge was given, and then their outpatient diary was made. After discharge, also for the follow-ups, outpatient diary was made. It is like there are two types. One is when the person is inpatient. Okay, when the person is in house, like in hospital, and we are doing it. We are randomizing them. We are giving the vaccine versus placebo here, and we are giving the challenges now, and we are following up the patient for what? We are following the patient for like the inoculation in the stool, where we check the clearance, where we check the clearance of the virus, or the percentage of virus we call it, or the the what we say, the presence of virus in the stool, right? Escherichia coli we can check through the stool. So we check after that during the discharge, during the hospitalization, and after discharge also we make the diaries for the patient's Escherichia coli clearance. Okay, if the patient is outpatient, simply we just give them the diary and we check for the time of inoculation strain output in the stools. Okay, for the Escherichia coli. So this is a simple thing. Okay, this is a simple thing, but very typical to, when you are reading that, it is very typical, and to make the entire protocol and entire thing, it is literally typical, guys. It is literally typical. So you must read these things and must understand. I can just tell you the basic things, but not everything. Okay. So this Escherichia coli example here. Then after that, regulations followed during CHIM. First is definitely if the development of challenge strain means development of the microorganism, development of organism, right? Development of organism can be done under the, I mean, it should be done under the good manufacturing practices. Good manufacturing practices. So CGMP must be followed, GLP must be followed, for reliable process according to good laboratory practices. Then GMP must be followed, like see, good GMP must be followed. International Council ICH must be followed. International Organization of Standardization ISO must be followed in case when we are developing the challenge in the company or factory or whatever the manufacturing units or the pharma companies. All these following things will be followed. And other than that, not only these, but the quality check is very, very important. Irrespective of the biosafety levels, there should be strict adherence to the laboratory design, personal protective equipments, biosafety equipments, implementation of SMPs, standard microbiological practices. SMP, how many of you have heard of SMPs before this class? How many of you have heard of SMPs before this class, guys? Tell me. Yes. Have you heard of SMPs before? No. Okay. So there are specific guidelines, like as we have the GMP, GLP, we have the ICH guidelines. There are specific standard microbiological practices for the development, check the bath, and to process the microorganisms in laboratories in the research. There are SMPs, standard microbiological practices. Okay. So SMPs, we must follow this. SMPs, we must follow the quality assurance for such studies. Okay. These are the major regulations. These are the major regulations we follow in the CHIM. Now, okay, so there are many more things. Ethical conduct of the research in CHIM. Let's learn from the guidelines. Let's check the guidelines now. I'll tell you from the guidelines now. Let me share the guidelines, guys. Okay, so this is how the guidelines looks like. Okay, this is how the guidelines looks like, right? So you can see Indian Council of Medical Research made this policy statement for the ethical conduct of controlled human infection studies CHIS. Okay, 2023. See, so first the ICMR bioethics unit, then who was involved, like the Roli Mathur, scientist F and head. So here you will see the, sorry, General Secretary of India, Government of India, Rajiv Behel, then ICMR bioethics unit head, Vasanta, Dr. Vasanta Muthuswamy, then all the, conveners, Dr. Gitanjali, chairperson, ICMR expert committee, and Dr. Roli Mathur, scientist F and head, right? So here are the things. First, they talked about what is this, then social values, the limitations, scope and objectives, technical considerations, responsible conduct of research, ethical considerations for such studies, and other things are there. Let me show you one by one. So I told you, so whatever, we have learned through the slides, right? Controlled human infection studies involve a research design that intentionally exposes healthy human beings to the specific pathogens. And these studies are, are aimed to access the vaccine treatment modalities and all, right? So these can be the applicable for all kind of, all kind of infections in all the developing and developed countries, in all the developed as well as the developing countries, for all type of infections, malaria, influenza, dengue, fever, or typhoid, cholera, whatever is there. These are for all the types of infections. Okay, so this statement means these guidelines is to be read in conjunction. As I told you, these are the part of ICMR National Ethics Guidelines of Biomedical and Health Research Involving Human Participant 2017 that we studied in the, that we discussed in the last class, right? So there are social values. We have already talked about social values of for the better understanding of the infection, and accelerating the product development of the disease, determining the minimum required of the dose of the drug, developing interventions, lower cost, de-risking the process by those things. So multiple things are there. Limited generability is there. Limitations. These are the limitations. right? Limited generability and short-term data, attenuated pathogen, that can be that not exactly accurating the behavior of wild pathogens. And then, there are usually sometimes this, endpoints are challenging, right? Methods have limited scope. So these are the limitations. Then scope and objectives, we already learned. Technical considerations. There are two types of studies. We have discussed these two types of studies. One is controlled human infections. One is controlled human infection testing. So one is CHIM, one is CHID. Okay, CHID and CHIM. Okay. So challenges strain must be developed in all the following. These are the regulatories what we must follow, irrespective of biolevel levels, like quality control and all must be followed for that. And there are other things also. There are other things also, like when we are importing the things and all, we must follow some guidelines. Study plans. So it's very simple. Study plans is a form of clinical research. One second, let me put the charts here. Yeah. So, like, the complete proper methodology must be there. If we are doing a clinical research, there must be the proper research design, methodology, adequate sample size, intervention, control group, rescue therapy, plan of standardized analysis. Everything must be properly written. Okay, everything must be properly written. So, CHIS commonly are coming under the randomized control trial. CHIS studies are usually the randomized control studies. Okay, CHIS are usually the randomized control trials, double blinding studies. Okay, double blinded studies, right? So the methodology should outline the required steps such as procedures for inoculating the pathogen, the range of dosing as applicable, laboratory investigations, what you have to do for monitoring, immunization or immunological assays, what you want to do, assessment of clinical outcomes, monitoring, any follow-up procedures. Everything must be done that is differing from pathogen to pathogen. So and about the, as we were talking about the complications and adverse event also, you should be very, very careful, right? You should have the place to address such complications like isolated wards, isolation wards and all, right? So again, there are the, different things like how to go for the scientific review. So expert review committee or expert independent review committee must be there. So these are the disease specific considerations. What are the considerations for malaria? Okay. And what are the considerations for dengue? Like test for the previous dengue exposure with rigorous screening, especially for frequent travelers. So there are specific guidelines for different parts like malaria, dengue, tuberculosis, typhoid. And then all it comes what we studied in last class, ethical institutional responsibilities, right? Responsibilities are always there, like closed settings, they require a closed setting. And then require the challenge stress is genetically modified, and the laboratory facilities and other facilities from the institution. Then review process, responsibilities of the researchers are given here, right? Collaboration and data that we studied previously, right? Then ethical considerations, like what is deliberate infection, right? Deliberate infection must have some rules and regulations, right? So it must have some rules and regulations. Selection of participants, selection of participant criteria within 21 to 60 years only. Guys, remember here, there is very specific thing. It is 21 to 60 years only. Participants should be healthy adults only. You should not, the kids and elderly patients are not allowed to participate in CHIP studies. This is a $1 million question. Okay. Anyone can ask, if someone asks you, what is the age group to participate in the CHIP studies? CHIP studies, that is 21 to 60 years, healthy adults. Okay. Unless otherwise justified. If you are doing in the pediatric, that should be justified properly. Okay. And the rigorous screening or health parameters must be done for that. Understand? So these are the specific guideline part. Then vulnerability we have discussed, benefits and risk we have to discuss all, talk about all those things. Additional safeguards like isolation wards and all must be there properly. Healthcare workers at the high risk, for the healthcare workers also there are guidelines. Family members of the visitors, that there must be all. Then, the informed consent, there are the parts of informed consent, what should be there. The basic elements of informed consent, statement about the research study, like others, purpose and procedure, duration of the participant, type of data collection and all. But here, there are some more specific things. Forcible risk, inconvenient harms, mitigation plans, and other than that, reimbursement and cost plans are different. Okay, and then the medical management is there. right? So these all things must be there in that. Okay, other than that, additional things are, no undue process, motivation to participant, psychosocial support, possibility of stigmatization, these must be, addressed in the informed consent form while taking for the CHIS study. Privacy and confidentiality, we all know. Payment for the participation, as we discussed in the last class. Study monitoring. So there are microbiology specific monitorings. Okay. Compensation, we have discussed that. Follow up of the participants, post sharing publications that we have discussed, ethical committee considerations. So here it is almost same of that, but here at least one or two microbiologists or independent consultants are important to be in the committee. Here that is specific, like one or two microbiologists or well-known experts must be there in the ethics committee. Okay, along with the normal ethics committee members, as we have discussed in last class. Then training of the EC reviews. So here all the simple things, foreseeable risk and all, they are defined here. Institution, participants, conflict of interest and all, right? So advocacy, publication, public interest, then research governance about the guidelines, like, should be NDCT must be followed, NABL must be followed, right? CDSCO must be taking care of that in India. CDSCO is the major part, like major regulatory authority, which takes care of the things, right? Then the references you will see. And at the end, you will see a fantastic thing, guys. After all these, annexes, you will see a fantastic thing here. And that is nothing else but the definitions. Okay, the glossary here is amazing. See, they defined the clinician, they defined the compensation, confidentiality, deliberate infection, ethical dumping. Rudely, you see, read about. Ethical dumping refers to the unethical practice of conducting research of experiment, often in the low or middle income countries, which would not be permissible or would face stricter ethical oversight in a high income or more developed countries. Understood? So ethical dumping, I told you, I explained, to you. So you must have understood. So you read these things and try to, explain by yourself what is legal expert, what is research related injury, what is risk, what is sponsor, stigmatization, negative, but perceptions about an individual because of perceived differences from the population at large. It could be because of the physical appearance, race, or sex. Okay, anything it could be. So stigmatization, test of understanding, a simple oral or written test designed to identify if the participant has understood the details related to his voluntary participation in the research before signing the informed consent form, right? Then vulnerability, everything is given here very nicely. Okay, and the people who are involved in that. So that's all for today, guys. And I think we have covered all these things about these guidelines. I'll share the guidelines. I'll share the, these study names also, what I've, and then the recordings. So you can read it and you can definitely, get the benefit out of it. And I recommend you all must read it, because when you are doing such studies, you are enrolled in such studies, you must know the guidelines, right? So that's all, guys. Now you can bombard your questions to me. Yep. Yes, guys, your questions, please. Yes, guys. Any questions? Ah, don't say no. You don't have the questions, really? Okay, Aditi is asking. Okay, fine, Aditi. We can talk about this. We can talk about this. If you want to publish the paper, we'll help you. Don't worry. Okay, we'll definitely help you, Aditi. Yeah. Anyone else, guys? Questions? Okay, so you all must fill the attendance form. Here is the attendance form, guys. Here is the attendance form. You must fill the attendance form, everyone. Okay. Mohammed Jaid is asking, sir, if a new company fulfills all the ethical requirements, can they start the infection studies? Definitely. Definitely. No, no, no. If they fulfill all the requirements, all the licenses of CGMP, CGR, GLP and all, they can definitely start. Okay. Details about double blinding. So see, there are three types of blinding. One is single blinding, one is double blinding, one is the triple blinding, we call it, or the complete blinding. Okay, we call it complete blinding also. See, there are the levels of participation in any clinical trial. There are the levels of participation. First level participant is who? The participant itself, the human participant who is participating in the clinical trial. Second level of participation is who? Second level of participation is investigator, the doctor or anyone who is investigator. The third level participant is the site. Okay, site managers or CROs, usually the CROs. Okay, third level is CRO, and the fourth level is sponsor. The fourth level is sponsor. So one blinding, the single blinding is when the person who is participating in the clinical trial is not knowing, he doesn't know anything about what he is taking. He is taking the drug or he is taking the placebo or control. Okay, that is called single blinding, when we hide everything about the drug and placebo from the participant. We just tell them like this is like for this disease, then this is the clinical trial drug. We cannot tell the details. So I mean, in the consenting time only, we tell everything about this, right? So there is single blinding. Double blinding means when neither the participant knows nor the investigator knows about what is drug, what is placebo. These comes only with the tag placebo drug or comparator drug. There is no details of the actual component or actual chemical entity inside, outside that. So you just have to give, okay, this person is randomized to the placebo group, give placebo. This person is randomized to the drug, give drug, but they don't know what is the actual chemical entity inside. So even investigator and if the investigator and participant both are blinded, both are, not knowing the drug, not knowing the chemical entity, that is called as double blinding. Triple blinding is when even the CRO doesn't know. Usually that is happening in very high end drugs when only the sponsor knows, but usually the things goes up to single and double blinding. Okay, understood? Raghavendran, Ravi, did you understand? Great. Anything else, guys? Any other questions? What are the actions taken when the trial in a healthy human volunteer doesn't go well and result in allergy or unwanted effects? Treatment of those. See, first is immediate discontinuation of the drug. Immediate drug continuation, drug discontinuation is the first thing. And second thing is treatment of the problem, treatment of the side effect. These are the two actions, being taken care by the people. So once these two things are, once these two things are covered, then everything is like, then everything is covered in this part, right? So first is discontinuation of the drug, and second is the treatment of that problem. Okay, and the actions are being taken by the ethics committee or by the legal team. If anything happens which is permanent or harmful, so they can apply for the compensation. The company, the sponsor, has to pay the compensation to the participant if they die, or if they have the permanent damage or permanent injury in their body. So that time the things are given. What if the drug is showing side effects only in a small community of people related to particular age or any particular racial group? So we usually discontinue drug, or at certain extent we check those things and the reason. And if we find that if the the testing drug is causing such kind of things, that is called as then we withdraw the drug from the community. We withdraw the entire trial from the community. If the entire community is showing the problems, or entire community or entire participant group is showing the problem, then we immediately discontinue the trial. We immediately terminate the clinical trial. Whatever money is invested, whatever things are invested, we have to immediately discontinue or terminate the clinical trial in such cases. What about the idiosyncratic effects? Okay, so sometimes idiosyncratic effects are there, positive effects are also there. In such cases, we find the drug more effectively, and we consider either escalating or de-escalating the doses. And also in such cases there are different scenario, what kind of effect the person is getting, what kind of effect the participant is getting. So we check that, and if the drug is too effective and drug is really good with the things, with the persons, with the race, with the gender or whatever, we check in the further indications with the further populations. Okay. So guys, if we are done, then we can wind up with the class, right? Thank you very much. Take care. I'll share the guidelines and the recording soon. Okay. Thank you very much. Thank you very much. Sir, as you said, there is a specific age for performing infectious studies in a particular age group. Then is there any loophole behind the vaccine development for infectious disease in children? Definitely. It is written here. If it is justified, like when we make the vaccines for children, we have to take special permissions. We have to show the specific safety parameters about the drug and all, like the low dose or usually the low dose drugs are checked in the pediatric. So according to the population, according to the population, we have to customize the doses, customize the safety parameters. We have to justify that, and we do that. Okay. Yeah. Okay, guys. So thank you very much. I hope you filled the form. So take care. Good night, guys. Feedback form was filled already, Harshita. You have joined late. Okay. I know many people are joining late. Many people are joining late just for the feedback form. So feedback form has been shared already for 5 minutes and this has not been given again. So no feedback form now. Please. Thank you very much, guys. Take care. Good night, guys. Good night. Take care.